asked by the Ministry of Health, Welfare and Sports to design a national collaborative structure for the development of new antibiotics. In 2016, together with the 12 Dutch organizations, he organized the potential of a new setup global information platform on AMR. In 2017, he founded AMR Insights and decided to focus his professional activities on combating the global threat on AMR. AMR Insights informs, educates, and connects professionals with the aim to curb antimicrobial resistance. AMR Insights has the ambition to develop into a global information platform on antimicrobial resistance. Maarten is committed to eliminating antimicrobial resistance because he does not accept the millions of innocent people need to die as a result of antimicrobial resistance. Therefore, here today, we have a highly established researcher. Dr. Maarten, we are so much honored to have you today, and welcome to our session today. Thank you so much. Over to you, Doc.
Thank you, Jimmy, for your very kind introduction. And also thank you for inviting me to contribute to this webinar series organized by CES Africa. I compliment you for the initiative of CES Africa, but also for this webinar series, because knowledge is extremely important when it comes to curbing antimicrobial resistance. I have a lecture of about 60 minutes, so I will end around 3 o'clock the local time. And it is an informal session, so don't feel free to interrupt me if you have any questions or remarks. Happy to answer. That can also be afterwards. I would like to share with you, well, I skipped the introduction because it was already done by Jimmy. I have an introduction about AMR Insights. I also want to tell you more about antimicrobial resistance as a global threat. Then I will start talking about key strategies to curb antimicrobial resistance, and then focus on the topic of today's webinar, which is antibiotic research, but also alternatives for antibiotics. And we also may have time for some questions and answers. Well, this is in following up on the kind introduction by Jimmy. I think it's important to mention that I took the decision 5 years ago to entirely focus my professional activities on curbing antimicrobial resistance. And this was for me a very principal decision point to decide to spend your professional life on adding to the curbing of AMR. And I've done now so for 5 years, and I'm very happy that I've taken the decision by that time. And also because I've been able to meet many inspiring and encouraging people around the globe. As next, I would like to tell more about the organization AMR Insights. Actually, what was already said in the introduction, AMR Insights focuses on informing professionals by means of a newsletter and by means of an information platform. We educate professionals by means of an AMR masterclass, and we connect professionals by means of organizing international matchmaking symposia and AMR innovation missions. And also, today's event is actually a webinar or a seminar on AMR. So this is part of our core activities. Last year, we took the decision to initiate a global network of ambassadors. And we currently are having 380 ambassadors in 55 countries. And these are professors, scientists, microbiologists, veterinarians, medical doctors, pharmacists, epidemiologists, and also many students, especially in the low and middle income countries. And we invite you to become a member of this network. And also Daniel Burungi and some others of you are also a member of this network. And we are also currently exploring possibilities to support student organizations globally who are involved in curbing antimicrobial resistance. Because of this powerful network, actually as a sort of a grassroots organization, we noticed an increasing amount of activities from this network, including many scientific publications, podcasts, projects, and also blogs. So these actually spontaneously come up bottom up, and they add to curbing antimicrobial resistance. AMR Insights focuses on different target groups. So not only on medical doctors and hospitals, but in fact, we do realize that many people in society, many professionals in society have to play a role, are responsible, have to play a role in curbing AMR. And these include, of course, the doctors in the hospitals, but also the people in the agri-food sector, in the healthcare, the animal healthcare sector, veterinarians and farmers, but also innovators in pharma and in biotech companies and also at universities, but also people in the environment. And also kids and carers have to play a role. And last but not least, also engaged students. So we have also set up a sort of a focal area group with a separate landing page for students who are committed to adding to curbing AMR. And in the next slide, you can see the landing page of this for the students around the globe. So this is a screenshot of the landing page, and here you can see that we will provide news and information especially meant for students. Was there a question?
No question, Doc. I think, yeah, the questions will be answered at the end of the sessions.
Okay,
Thank you,
perfect. Then coming to the project of AMR Insights and the ambassadors. Actually, I would like to just briefly mention some of our projects. One is we have set up a Dutch consortium, Antibiotics from Water, and this consortium is focused on the reducing the discharge of antibiotics in wastewater from hospitals and manufacturing plants. And we have also recently produced a white paper, which you can see on the right-hand side. Then we are in the process of developing a serious game meant for medical doctors. And this serious game has the objective to increase the awareness on AMR for medical doctors. And this will also be released in the low and middle income countries. Then we have organized on 24 November, at the last day of the World Antimicrobials Awareness Week, what we call AMR Relay event. And this was a 24-hour event with 96 time slots with organizations around the globe who are involved in curbing antimicrobial resistance. And another project we have initiated is that we are currently carrying out a sort of a multi-country retrospective clinical study where we look at the misuse of antibiotics in hospitalized COVID patients in 7 countries. And as you all know, COVID patients, corona is a virus, so antibiotics do not work against corona, but in practice, many medical doctors do prescribe antibiotics for COVID patients. And we want to estimate how serious this problem is. Then another project we have initiated 2 years ago that we have set up a sort of a technology database for technologies who can be used to curb antimicrobial resistance. And this is a technology example by the company Evotec, which is an international drug antibiotics developing company. And related to this database is also a very dedicated filter, which we have developed such that you can easily find the proper technologies when you're looking for certain technologies on the basis of application, microorganism, partnering opportunities, and so on. This is about AMR Insights. Then I would like to go to antimicrobial resistance, which is, of course, as we all know, a global threat. And fortunately, it has come in the public domain. So it is not only a problem for scientists and medical doctors, but increasingly also normal people, citizens, patients start to know more about the problem of AMR. And we increasingly do realize that AMR is a threat. So something must happen, and preferably now. And your initiative, South Africa, is a very good example of this. Antimicrobial resistance is a very complex and abstract problem. There's a lack of understanding. It is difficult to explain to other people what antimicrobial resistance in fact is. And there are many data about AMR, but there is no overview. And if you compare this to corona, where you can see on the right-hand side the screenshot of Johns Hopkins University, where they give sort of a very, very detailed overview of the amount of casualties of corona. And this is all updated on a regular basis. And such an overview is not available for AMR. So we only have very rough estimates, like 700,000 deaths per year, maybe more. But where and how and when exactly, we do not know. So we also are not able to see trends and developments. Because AMR is a difficult problem, I often use 10 statements to explain AMR to people in my lectures. And I will now go through these 10 statements. First of all, is AMR is a global threat. It can affect anybody, everywhere, at any time. Men, women, healthy people, sick people, young people, old people. And there's sort of a continuous spreading of AMR around the globe because of the traveling of people, traveling of shipment and transport of meat and animals. And in fact, this is because microorganisms can spread around the globe, bacteria, but also genetic material, plasmids, can spread around the globe, and also antimicrobials, such as you can see here, this chemical structure. Also, these antimicrobials can spread around the globe. So, in fact, because of these three aspects, there's sort of a continuous movement of resistance around the globe. And as an example, when, for instance, in China, a certain resistant bacteria is identified, then it may be 1 month later also in India, and then maybe a couple of weeks later also in Africa and the United States. So it is spreading on a continuous basis. The second statement is that AMR is a hidden threat. It is not always diagnosed, or in many cases it is not diagnosed. It is not documented always, and it is not always such that the data are being collected. So this is also related to an earlier statement. Actually, we do not know very much about AMR. And this is a problem because if you don't know the statistics of AMR, you cannot measure also the impact of interventions because you simply don't know how many there were in the beginning and how many there were after the intervention. So there is still a lot of work to do. We should come to a much better statistics of AMR and more precise, and it should be better diagnosed, it should be better documented, and data should be collected in global databases. Another aspect is that, one second. AMR is not only applicable to bacteria, and we all know that bacteria can become resistant to antibiotics, but in fact also other microorganisms, including viruses. Look at the coronavirus, because we see all these new variants, and in fact these are all resistant variants of the original coronavirus. Also yeasts, fungi, and parasites can become resistant. So for that reason, we also talk about antimicrobial resistance and not only about antibiotic resistance, which applies to bacteria. Overall, roughly speaking, there's a correlation between the use of antibiotics in a certain country and the amount of AMR observed. And this is an older picture of the review on antimicrobial resistance by Jim O'Neill in 2016, if I'm well. And there you can see, at least I hope you can see it, that in countries like France and Spain, people used much more antibiotics than in countries like the Netherlands and Sweden. So overall, in France and in Spain, you also see much more AMR than in the Netherlands and in Sweden. And so there's every reason to decrease the amount of antibiotics, because this will also result in less resistance. At the same time, we must realize that banning antimicrobials is not a solution. We desperately do need antibiotics. And this is a very simple graph where you can see, the more antibiotics you use, this may result in resistant bacteria because of the high amounts of antibiotics you use. But at the same time, if you lower the amount of antibiotics, you go to very no prescriptions or hardly any prescriptions, then the result may be that you get infectious diseases coming up again, and this also, in fact, may happen in some of the low and middle income countries where some antibiotics may simply be not available or not affordable. Globally, roughly speaking, AMR is escalating. The problem is becoming increasingly serious. And as you can also see in this red line, and at the same time, we notice that the number of antibiotics in the pipeline, because we do need novel antibiotics, we will discuss that later on as well, is going down. So the pipelines are becoming increasingly empty, and it is increasingly difficult also to develop and commercialize novel antibiotics. At the same time, we do need these novel antibiotics very much. I will come back to that later. AMR has an uncontrollable dynamics, and that is for three reasons. First of all, bacteria can divide very rapidly. And the second point is that AMR can arise also very rapidly. And the third point is that AMR can easily be translated from one bacteria to another. And I can show you that here, when you go to bacteria, they take about 20 minutes to divide. And so you can, this is of course very short.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacteria, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
So the bacteria divides. And the second point I mentioned is that AMR may arise very rapidly. I have a short video here where you can see how bacteria, who were originally sensitive to all microorganisms, became resistant in just a matter of a few days to very high concentrations of antibiotics. And I'm not sure, can you hear the sound of the video?
Yes, Doc, yeah, we can hear it.
Okay. Now,
so what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands. And at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, porous and thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in an area where there's no antibiotic, up to the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
The video shows that what was said, that bacteria can become resistant to extremely high concentrations of antibiotics. The third video shows, and I will not show it here, but shows that also when bacteria are resistant, they can very easily transfer these mechanisms, these resistant mechanisms, to other bacteria. I come to the next statement, number 8, that is AMR makes also medical interventions impossible, such as cesarean sections, other surgeries, but also chemotherapy and stem cell therapy. And this is because these medical interventions do need the routine use of antibiotics as a preventive to avoid infections in these patients. And if the antibiotics are not active anymore, then these medical interventions cannot be carried out anymore. The 9th statement is that AMR, antibiotic resistance, is observed with all commercially available antibiotics. So it turned out that all antibiotics that came on the market since 1930 of the last century, the previous century, resulted in resistant bacteria. And sometimes after a week, sometimes after 2 months, sometimes after a year. But for all these antibiotics, resistant bacteria were observed. Then I come to the 10th statement of AMR, that is that there is no one single solution to curb antimicrobial resistance. And what you can see here is the comparison between AMR and, on the right-hand side, climate change. The climate change is comparable in the way that there is not one single solution or different solutions. And the same applies to AMR. And what you see here on your left-hand side is the picture in the famous report by Jim O'Neill, where he forecasts that in 2050 we may have until 10 million deaths per year following antimicrobial resistance. So it is a global threat, and there's every reason to work hard on trying to minimize the very scary results and effects of AMR. To do so, we do need to curb AMR. And in fact, AMR Insights uses 6 different key strategies as a starting point to combat AMR. And the first one is that there's a much better prevention of infectious diseases by means of vaccination, but also by means of wash, with the water and hygiene. And the second one is that we do need a much better stewardship in the human and in the animal health care. The next one is that we do need a discharge of antimicrobials, less discharge of antimicrobials in wastewater and from hospitals and from manufacturing plants. And the fourth one is that we do need better diagnostics, affordable, cheap, rapid diagnostics, which can easily distinguish, identify bacteria, but also determine if bacteria are resistant or still sensitive to certain antibiotics. Then we do need novel antimicrobials, as will be discussed later on, and we also do need alternative antimicrobial therapies, such as phage therapy. And also this will be discussed later on in this presentation. I will now come to antibiotic research, because this is the main topic of this lecture. And now start talking about, what it in fact means when you start with research development of novel antibiotics. And in fact, as with many other pharmaceutical products, there's a sort of a chain of activities you have to carry out in order to investigate if a potential compound, a small molecule or a compound, could indeed be developed until a marketed product. And this is a lengthy project. It may take up to 12 or 15 years. It may be, it is extremely expensive, $1.5 billion or even more. And It is complicated. You do need many expertises, and I will also share some more insights on R&D of novel antimicrobials. And this is, in fact, what you can see here is the rough steps to be taken. Basically, the pre-hit to lead. Lead compound is a compound which looks promising as a starting point for a final new antibiotic, but also lead optimization, preclinical, clinical studies, and later on the regulatory review, where the regulatory authorities will just examine the dossier in order to see if indeed the antibiotic can be registered or not. And even after approval, there may still be some additional research to market surveillance studies to investigate the safety and efficacy of the product. Roughly speaking, the pre-hit to lead work is done by academic institutions and paid by academic institutions, and sometimes also by governmental institutes and by the EU here in Europe. And what you can see here is that typically the pre-hit to lead work starts with many compounds which you investigate, hundreds or thousands of compounds, and then at a certain point in time you come to the next stage, hit to lead, lead optimization and preclinical, where the funding is taken over by organizations such as CARBEX, the Repair Impact Fund, and the AMR Action Fund. And typically, when you come to this point in time, the number of compounds goes down to 1 or 2 compounds because of the selection process here. So, because it would simply be impossible and also too expensive to test all the compounds which you have initially tested. Most likely, or in most cases, 1 or 2 or several compounds pop up here, and they will then be further investigated from hit to lead, lead optimization, and preclinical studies. What I said, this is primarily done by academic researchers, institutes, sometimes also by pharma companies when they have their own research and development organization. And then this work is mostly done by startup companies and SMEs, so small companies who are flexible enough to work with these lead compounds and develop these to the next stage. And what I've said for the last phases are mostly done by pharma companies because this is extremely expensive, and mainly because of the expensive clinical trials. So actually here you start really spending the big amounts of money, and only pharma companies have deep pockets and can afford to pay the clinical investigations. I will also tell you a little bit more about drug discovery and development when it comes to what activities are being done in these stages. And what I said, this is pre-hit to lead. This is done by academia at the pre-hit to lead, and then this is mostly done by startup companies, SMEs, and hit to lead, lead optimization, preclinical. And what you see here, these stages are, for instance, structure activity analysis to compare, for instance, what does the chemical structure say about the activity, but also in vitro tests and proof of concept, synthesis route, how can you synthesize the compound. Then we come to the lead optimization, and there new analogs may be tested, but also some microbiology tests to test the effect on microorganisms, some in vivo toxicology with test animals, but also some in vitro safety and in vivo activity. And you can understand that along this value chain, the companies get an increasingly better understanding of the compound and of its characteristics. Then we get, before the clinical trials, we do get the preclinical trials. And this is again, microbiology, animal studies, and also importantly is to focus more on how can we in the future manufacture the antibiotic at a small scale, at a medium large scale, but also ultimately at a large scale. And because this is also important for the production of the compound. After these stages, there's sort of a decision point, and where the pharma companies just analyze all the data obtained. Well, is it promising? Is the antibiotic not too toxic? Is the antibiotic efficacious enough? Are there no side effects? Can it easily be produced? And on the basis of these criteria, it may be decided to enter into the clinical trials. What I showed you before, the clinical trials are mentioned here. And these are well divided in 3 phases. Clinical trials, phase 1, 2, and 3. The first phase, phase 1, is a small study in human volunteers, healthy young people. They are just tried as sort of a human test animal to look at the first effects on a human being. Then the second trial, clinical phase 2, is to investigate the dose efficacy relation. So it is just investigated what doses are needed to make the antibiotic efficacious in patients. And the third phase is that you, at a very large scale, investigate the selected dosage. This, what I said, the clinical trials are by far the most expensive, hundreds of millions of US dollars, and they take many years also. And the challenge for companies, of course, is not to start these clinical studies if there is some doubt, because it may simply be too expensive to start clinical trials if there is some doubt, because then it may be just a waste of money. So I was focusing first on the hit-to-lead optimization and preclinical phases. I also would like to zoom in on the very first phase, which is mostly done by academic research center. Because what is done there is really the basic research and the exploratory antibiotic discovery. And this is mostly very, very fundamental research, chemical research, and also chemical synthesis work, the use of databases, things like that, especially a lot of chemistry. Then I would also like to zoom in on another aspect of the first phase of academic research. You're medical students, so academia is an important arena for you. And in fact, when you are working in this pre-hit-to-lead phase, which is the very first academic phase, then already a lot of expertise are needed to do this work. Like you need to know how to culture bacteria, how to screen. You need to know about combinatorial chemistry, medicinal chemistry, but also bioinformatics, structure-based design, biological profiling, modes of action, antibiotics. And there are also many skills are needed in this phase. Because team building, you have to collaborate with different disciplines. You need to be able to manage complex processes, but also you need to know the regulatory requirements later on, drug development process, the costs involved, and so on. And last but not least, also as mentioned here, this is an important one as well, GXP. The work, in fact, needs to be done according to good research practice, good laboratory practice, and good clinical practice. So you must also be aware of how these guidelines work and how you can also follow these up. Then there's also expensive infrastructure needed in the academic institutions, such as HPLCs, animal facilities where you can have your test animals, but also compound repositories and compound libraries, radioactivity labs, and for radioactivity research. So you can imagine that there are a lot of expertise, a lot of skills and a lot of infrastructure are needed. This is what we call capabilities. Okay? You have to be capable to do so, and this is what we call capacities. You have to have the capacity to carry out this work. Okay, so I told you something about the challenges of the research and development process of novel antibiotics. There are also many pitfalls, and I would like to mention a recent publication which was made by a team of writers from the AMR Insights Ambassador Network. And this article was published in Microbiology Research, and the name was Looking for Solutions to the Pitfalls of Developing Novel Antibacterials in an Economically Challenging System. And I definitely recommend you to download this publication. It can easily be downloaded from this URL. Then you can directly get the PDF of this document. And in fact, this document shows the different challenges and the pitfalls. And I don't want to discuss it in too much detail, but I would like to again mention once again that the pipeline is very empty for novel antibiotics. As you can see here, that the number of new antibiotics in the past decades was especially high in the 1950s, in the 1960s, and the 1970s. But since then, actually, we have not been able to come up with an entirely new antibiotics, or only some kind of variations on chemical structures. So we do need novel antibiotics desperately. And if you're in the process of developing antibiotics, then you're faced with challenges such as, can you set up a business case? Can you get the return on investment? Can you get the funding? But also, are you able to deal with the science? Do you get industrial support from pharma companies? It is not very sexy to work on antibiotics. It's much more sexy to work on oncogenic projects. We are facing that many people who are experienced in pharma R&D are leaving the industry, so we may also lose their expertise, and there are many other challenges. So it is it is a pretty challenging domain, and at the same time, what I said, we do need novel antibiotics. So I do not want to discourage anyone who would like to get involved in this business. Okay, then another aspect is also what I told before. There's more than only antibiotics and antimicrobials. There are also many alternatives to antibiotics, and I think one of the most famous alternatives is phage therapy, but there is much more, in fact. We are in the process of writing a new paper, again by the AMR Insights Ambassador Network, and this paper will hopefully be published early coming year in the journal Antibiotics, and that is an overview, a review of the alternative strategies to combat antimicrobial resistance. And as you can see, there's a large team of ambassadors is involved, and from many different countries around the globe. And it is very special that so many ambassadors in different countries are able and willing to join forces in writing teams to generate such these scientific papers. And you must also realize that these people have never met each other before. They have never seen each other. They only have, they started to work in sub-teams on these, in this case, on the alternative strategies. I would like to go to the next slide, where I have just shown you a selection of alternative antimicrobial strategies. So what you can already see is that there are many different ways to combat infectious diseases without using antibiotics or other antimicrobials. And I would like just to mention a few of them, because it is simply too long to go through this entire list. But as an example, as you may go for a combination therapy with antibiotics and other substances. Sometimes you can also use biocides. You can sometimes use natural products like essential oils and substrates of medicinal plants, especially also in countries like India. And I already mentioned the bacteriophages. They can be used, but also enzymes produced by bacteriophages can be used to combat bacteria. Then there are also nanoparticles which can be used, but also antimicrobial peptides and non-mammalian non-nucleic acids. Then there are also very sophisticated drug delivery systems, molecular reprogramming, biofilm dispersion, and also the use of antivirulence compounds, which make bacteria less infectious. So there's much more biofilm agents, monoclonal antibodies, important aspects against pathogens, and also the use of vaccines, what I already mentioned in the beginning. You also see here the word escape pathogens, and escape is an acronym initiated by WHO, the World Health Organization, which clearly indicates the most important resistant bacteria, or most are resistant, where we do need novel antibiotics. Well, to continue RNA DNA vaccines, but also reverse vaccinology, CRISPR-Cas, novel genetics technology, and more. So the reason I show you this is that we are not only dependent on antibiotics and antimicrobial compounds, but we can also make use of alternative strategies, such as you see listed in this slide. And it is not that we can say, well, it is one or the other. We do desperately need both of them. And what I said, the AMR problem is increasing at a global scale, and I'm convinced that we also should increase the number of alternative approaches, bring them to maturity, bring them to the market, such that they can be easily used in an affordable way around the globe, and especially in the, of course, lower middle income countries. This is what I have wanted to share with you. So I've told you a little bit about myself. I have also informed you about AMR Insights. What AMR Insights is doing is a globally active network-based organization. I also mentioned it's AMR Insights Ambassador Network, and what I said, I welcome you if you're interested to become an ambassador. I've informed you about antimicrobial resistance, and in these 10 statements. And I've also indicated that there's not one single solution to curb AMR, but that we do different strategies. And then I informed you about, well, what it takes to develop a novel antibiotic and the complexities and challenges related to that. And that we not only do need novel antibiotics, but we also do need alternative, such as phage therapy and one of the other therapies and alternatives mentioned. Okay, I hope this my presentation has been clear. I hope I've convinced you that we do need to join forces in order to curb AMR. I hope I have also indicated that we welcome students to get involved in this global curbing of AMR by means of the AMR Insights Ambassador Network or in any other way. I am happy to take any questions and give the floor back to Jimmy.
Thank you so much, Dr. Maarten. Actually, we have learned a lot. Very informative, actually, and it is very educative. In this section, I think I will offer anyone who have a question, before we proceed on. Thank you. Oh, I think, yeah, I think maybe we don't have anyone with a question. Maybe Doc, I can just ask one of the question, maybe, yeah, to clarify on maybe what may be the importance of technology database information as far as drug research.
Okay.
Can you repeat the question?
Okay, yeah, as far as antibiotic research and discovery is concerned. Over to you, Doc.
Yeah. And what is your question on research and development of antibiotics?
Yes, the importance of data technology information as far as, yeah.
No, I got you, sorry. That's an excellent question. We increasingly do need data technologies to curb antimicrobial resistance, especially also in research and development. And the use of databases with chemical compounds, but also data technologies to use, for instance, in surveillance of AMR around the globe. I can tell you that every year in June, we are organizing an event which is called Advancing Data Technologies to Corner AMR. And this event focuses on how can we use data technologies to curb AMR, antimicrobial resistance. And as of that, it is clear that there are many different data technologies that can be used, including, for instance, artificial intelligence, machine learning, deep learning, but also Internet of Things, blockchain technology, internet technology. And we have taken the decision earlier this year to draft a policy paper on the use of these data technologies when it comes to curbing AMR. And we are currently writing this paper together with a team of co-writers, co-authors, again in many countries, where we zoom in on how can we now use data technologies in research and development, but also in surveillance of antibiotics and in stewardship programs. So, and I'm happy that the topic does get an increasing amount of attention.
Thank you so much, Doc, for elaborating the question. Maybe, yeah, we have. I think we have one question. Yeah, maybe you can just offer this chance to anyone with a question. I think I had someone, yeah, who wanted to ask a question. Over to you.
Okay, thank you so much. I would like to thank Doctor for the wonderful presentation.
Okay.
Okay, Mike could be the schedule for how we are going to be learning, but I don't know like how we are going to be actually communicating, getting this information such that we also prepare to fit into the schedule. How are we going to do that? Yeah, that was my question. Like, are we going to be having a platform where we shall be communicating from, or we are going to use the previous platform? Thank you.
There's a question to me.
Yeah, I think that question goes to me. And yeah, we have a WhatsApp group that is, we used to organize everything, the activities that are ongoing, the webinars, and also other activities around. Maybe, yeah, I will share the link for the WhatsApp group. Maybe whoever who is not in the group can easily join the group. Thank you so much. Yeah. Maybe, yeah, Doc, I'd like to ask you maybe one question. Maybe, yeah, as far as, yeah, these are the young students, enthusiastic researchers, yeah, who want to get into research and development of antibiotics and antimicrobial resistance in particular. In general, sorry. So maybe, yeah, how can you, yeah, what's your thought? How can we empower these the young researchers, yeah, to be innovative, also, yeah, to bring them and train them into this field of drug research and development? Okay. Over to you.
Yeah, good question, Genji. First of all, I would say follow many online courses on research and development of novel antibiotics. There's a lot of information available, also on YouTube, but also many organizations offer very informative videos and webinars. An example is also GARD-P. I don't know, I will just put it in the chat, because it's important that you are aware of GARD-P. Let me just see if I can find the chats here. You should definitely look these up. And there's also GARD-P Revive. And these organizations offer videos, information for researchers, but also a network of experts who can easily be approached with questions on research and developments of novel antibiotics. So I'll just again add it also in the chat. They also have experts. So I would definitely contact these organizations, these people. That's one. The second advice I have is to young researchers is to get connected, especially from Africa, get connected to research groups around the globe and research initiatives with antibiotics research. And because a lot is going on and it is complex, so you do need to have possibilities to get to get interaction, to learn, to collaborate. And so that is also very, very important to get connected, also by means maybe of master classes and subscribe to newsletters. And also there are quite some organizations who are involved in supporting initiatives on novel antibiotics R&D. And just as an example, but that is then for Europe. We have this organization INCATE, a new organization in Switzerland. They're focusing on novel upcoming antibiotics. As an example, you also have the AMR R&D hub. And there are many organizations. So coming back to summarize this, so one is follow training courses, attend or look at YouTube videos and contact organizations, which is what I just mentioned. Get involved in projects. Maybe also see if you can do your PhD in a European or a US university or medical center. Also investigate opportunities to collaborate with companies where the experience is available. So a long answer for a short question.
Yeah, absolutely. Yeah, fantastic. Thank you so much, Dr. Yeah, maybe, for example, yeah, GARD-P actually is one of the organizations that actually they have been of great help to us. We have been contacting them. They have been contacting us. They have very nice resources for learning. That's we have been using them to extract some information which have been of great help to us. Maybe the organization, the webinars they have been organizing, they have been of great help, and actually GARD-P is a very nice organization. It is thank you that you have referred it to us, and and many more other organizations. We will be happy to have many others like GARD-P and also Insights, AMR Insights if possible, and we will happy to have more support from you and also from other organizations which you may be of contact with, as we strive against this global health threat of antimicrobial resistance. And because actually, this is a very big challenge that is affecting the world globe, and research is a very nice field to innovate actually effective solution against antimicrobial resistance. But we believe that without research, actually, it could be difficult to manage this problem. We would support, maybe, we would appreciate any support from any other organization, especially from AMR Insights, which you lead. Actually, that's we really appreciate. Maybe, without checking.
Just to interrupt you. AMR Insights is most willing to support and to help. We have a free information, a free newsletter. You can subscribe. We have an information platform where you can also, with there's a search bar. And if you type in, for instance, discovery, or you type in Africa, or you type in Kenya, or you type in animal health, then you will get a selection of all the news and information on that topic in relation to antimicrobial resistance. So if you type in the search bar Kenya, you'll get, well, maybe 20, 30, 40 recent posts on AMR in Kenya, as an example. And also what I said, I welcome you to join the AMR Insights Ambassador Network, because the Ambassador Network is expanding, is growing, and there are many committed people, and including from your organization also, Daniel Ruby. And what I said before is we are in the process of now also to work out, to develop a sort of a collaborative structure between the different students' organizations, such that they can well effectively also collaborate and exchange information. And last but not least, I also want to compliment you really for setting up SAS Africa and the great work you are doing. My compliments. Keep on working on this excellent goal. And I wish you every success, and happy to stay involved in the future.
Thank you, Doc. Thank you, Dr. Maarten. Yes, as I have said earlier, yeah, maybe I want to emphasize on the program that is ahead of us. That is the French program on drug research and development. And yeah, for those students who may be interested to join the program, the same information will be conveyed in our WhatsApp, in the subgroup, whereby the students who may be interested to join the program will be required actually to start the program early next year. That will be a program that will take actually around 5 months. And it will be an in-depth learning to introduce these students to the process of involved in drug research and development. And I'm happy that some of you, many of the students have actually showed interest in the same program. And I will do request that maybe Dr. Maarten, whenever we call upon you to assist in that upcoming program, maybe be ready. We requested that be ready to help, and also to provide maybe a sourcing of other speakers in some of the selected topics, which will be shared later with you. Thank you so much. And without taking much time, I would like to offer this chance to Dr. Maarten to give us the last words before we end the session. Over to you, Dr. Maarten.
Okay, well, thank you. In fact, I want to thank you all for inviting me. Keep up the good work. And I look forward to intensifying the collaboration with your excellent organization. Thank you.
Thank you so much, Dr. Maarten. We're so much grateful for having you today. This session has been very informative, very educative, and I'm sure everyone in this session have picked an important project that will be of great help in antibiotic research and discovery. Because actually, as you have provided the alternatives for antibiotics, they showed the potential of coming up with new innovations against AMR. Thank you so much, everyone, for attending this session today. And we would like to request you to be active in our previous sessions, which will be, I think our last session will be next Saturday, and the same information will be communicated in our WhatsApp group. Thank you so much, and I wish you a wonderful time ahead. Thank you so much. Bye-bye.